Now we're going to move on to Fimbriae, okay? And so these are fine, proteinaceous, hair-like bristles that emerge from the cell's surface. Proteinaceous just means protein-based, okay? And these are going to function in adhesion or gluing, sticking to other cells and surfaces. So here is a picture of Fimbriae. You can see that it covers the whole entire surface, and they're kind of cool, okay? This is what they look like up close and personal. You can see the fimbriae are here next to E. coli cells and the internal microvilli. So this is what it would look like in real life. So then we have pili, and these are rigid tubular structures that are made of pilin protein. And these are found only in gram-negative cells. So that will be important. So when we do things in the lab, if you see a pili, then you know it's going to be gram-negative. And these also function to join bacterial cells for partial DNA transfer called conjugation. So here is a picture of, again, the little tiny guys are fimbriae and the pili are the big guys. Glycocalyx, this is where we have a coating of molecules external to a cell wall, and they're made of sugars or proteins. And there are two different types. There's a slime layer or there is a capsule. And the slime layer would be loosely organized or attached, and the capsule would be a highly organized, tightly attached type. So here's where it looks like the slime layer that literally looks like slime, or we can have the capsule. So what is the function? Well, the function is to protect the cells from dehydration and nutrient loss. And this inhibits killing by white blood cells by phagocytosis, which is that Pac-Man one. And it also contributes to pathogenicity. And it also can help us with attachment by the formation of biofilms. Okay, like the slime on your teeth in the morning before you brush your teeth, that would be a biofilm. So here is a picture of a colony without a capsule. It's kind of just where the colonies with a capsule are more organized. Okay. So what about functions? Well, this is another picture of capsule versus the cell bodies. Okay. So biofilm, like I said, I use kind of like the teeth example, but we can also have biofilm on a catheter. And so this would not necessarily be the best thing, right? So this would be like biofilm matrix, the cells of the biofilm, and then the surface of the catheter in which they would attach and grow. So moving on then to the cell envelope. Sorry, I'm taking notes so I can label the slides as I upload them. Okay, so the cell envelope, this is the external covering outside of the cytoplasm, and this is composed of two basic layers. We have the cell wall and the cell membrane, and this maintains cell integrity. And there are two different types of bacteria that are demonstrated by the gram stain. This is going to be gram positive, and these have a thick cell wall composed primarily of peptoglycan and a cell membrane, and where we have gram negative bacteria. And this would have an outer cell membrane, a thin peptoglycan layer, and cell membrane. So this structure of a cell wall is going to not only determine the shape, but it's also going to prevent breakdown or lysis due to the changing osmotic pressures. All right, so this is good. It kind of keeps them alive, right? And peptoglycan is the primary component. And this is a unique macro molecule, meaning macro being big, composed of repeating frameworks of long glycan chains that are cross-linked by short peptide fragments. So the figure of the structure of cell walls that you can look at in your book is where we have the peptoglycan of a cell wall, and this is a huge three-dimensional lattice work that is actually just one giant molecule that surrounds and supports the cell. And this shows the molecular pattern of peptoglycan. And it has alternating glycans, such as NAG and NAM. And these would be bound together in long strands. So the NAG stands for N-acetylglucosamide, where the NAM stands for N-acetylmurmuric acid. And then adjacent murmuric acid molecules on parallel chains are bound by the cross-linking of the peptides. And so those would be the brown spheres on the image in your book. Okay. And then we also have an enlarged view of the links between the NAM molecules And this would be where we have a tetrapeptide chains that would break off the muric acids and that are connected by amino acid interbridges. And the amino acids in the interbridge can vary, or they may be lacking entirely. And it's this linkage that provides the rigid yet flexible support to the cell. So here is your pictures where you have the NAMs and how they're attached, and very, very close up, middle close up, and an outside view. Okay. So what about a gram-positive cell wall? Well, this is where we have 20 to 80 nanometers thick peptoglycan layer. And this includes the teichoic acid as the lipoteichoic acid. And these function in cell wall maintenance and the enlargement during cell division. They also move those cations or those positive charged ions across the cell envelope, and they can stimulate a specific immune response. And some cells have a periplasmic space, and this would be between the cell membrane and the cell wall. Okay, so here you have your gram-positive. This is a great figure. You have your peptoglycan, you have your teichoic acid, your phospholipid, your membrane proteins, right? And so all of the keys there so that we can see the gram-positive wall. Well, then what about the gram-negative? Well, this is where we have the inner and outer membranes in the periplasmic space, and between them we contain a thin peptoglycan layer. And the outer membrane contains the lipopolysaccharides, and this is the lipid portion, or the outside toxin, and this may become toxic when released during infections. We also have this that can function as receptors, and it can block the immune response. And these contain porin molecules in an upper layer, and these regulate molecules entering and leaving the cells. So here's a picture of your gram positive, right? Or I'm sorry, your gram negative cells. Right, so here's the comparison then of gram positive versus gram negative. And then also a fun little table of comparing positive and negative. So this would be a great one to print off and to really, really, really learn. So let's do a concept check. So which of the following is not found in a gram negative cell wall structure? Pause me and come back. All right, the answer is B, teichoic acids. All right, great job.